course now, moving from the parasites into the study of the fungi. First and fundamentally, fungi come in macroscopic forms, they come in microscopic forms. Macroscopic fungi include mushrooms, which is why I have the cute little picture here of the mushrooms. Mushrooms, we eat them perhaps, but they don't cause infection. So when we study fungi in a mycology class, we're going to study exclusively the microscopic fungi. So this is your first and last look at a mushroom, unless you're going to fix some for dinner tonight. The fungi have a eukaryotic cell structure, just like us, meaning that they have a true nucleus and cellular organelles. They do not have chlorophyll, but rather they absorb all their nutrients from the environment. Microscopically, we divide fungi into two groups, the molds, which have hyphae, and the yeasts, which are more typically oval budding cells with no aerial hyphae. Are characteristic structures found in molds. When you look at them, they're kind of somewhat reminiscent of roots, if you will, on a plant. They're not roots, they're hyphae. Um, it comes in a couple varieties. One is called aseptate hyphae. A, of course, means lacking or without, so lacking septum, or non-septate is sometimes used by individuals. So the septum is like a crosswalk. And down on a cellular level, it's a demarcation of where these cells are dividing. So when it comes to the aseptate or non-septate hyphae, there's no cross walls. They tend to be wider than the septate hyphae. They're often described as ribbon-like. I don't know, that has never particularly made an impression on me of reminding me of ribbon, but it may for you. The septate hyphae then has these cross walls. They're not always terribly easy to see. So even though we have nice sketches here where they stick out and you couldn't miss them, do know that, when you're actually peering at them through a microscope, sometimes you have to look kind of hard to see them. They tend to be narrower than the aseptate hyphae. When hyphae is down below agar surface, when you're growing it in the lab, we refer to that as the vegetative hyphae. When it's down below the agar surface, it's absorbing the nutrients from the agar. On the other hand, the aerial hyphae is what sticks up above the agar. This is typically where we find reproductive structures. So if we're specifically searching for them in a fungus culture, we want to concentrate on looking at some of that aerial hyphae. Phyla in the fungi that we should be aware of, and they're separated based on the sexual reproductive structures of a given fungus. So first of all, we have the zygomycota. These do have aseptate hyphae. The sexual stage we call the zygosporangium. So you can see the tie-in between the zygomycota and the zygosporangium. The ascomycota have ascospores in their sexual stage. So ascomycota, ascospores. The basidiomycota have, you guessed it, basidiospores. And then lastly, the deuteromycota, this poor guy doesn't have a sexual phase, and at least not that we've discovered yet. So I am, in terms of taxonomy, I think the most important takeaway from this is as we work our way through these, we're going to use these terms, ascospores, basidiospores, etc. And so now that we realize that these sexual stage structures have been named with the name for the phylum, then it makes a lot more sense that way. Another way to consider dividing various fungi from one another is based on whether they have a sexual stage or not. If they lack a sexual stage, they will reproduce by something called conidia, and we're going to look later on at organisms with characteristic microconidia and macroconidia. If they have a sexual stage that's been identified, um, then they fall in this other group. And I just think it's always a little bit comical to look at the way they were named originally um, by the the guys, the men who first did the research and the naming, and that if a fungi had a sexual stage, it was considered perfect, and if it lacked a sexual stage, it was considered imperfect. Let's bring up some important points about specimen collection for fungal cultures. Of course, it's the doctor that's going to be collecting the specimen, and the picture that you're looking at in the lower right is a doc collecting what is called a skin scraping from the bottom of the patient's foot. In his hand is a scalpel blade where actually digging down um, to the dermis level in order to collect the tissues that he thinks may be infected with a fungus. It's important to us in the lab that the doc sends a sufficient amount of specimen. Often we're going to be plating fungal cultures on multiple plates and tubes, and so we have to divvy it up and put a sufficient amount of inoculum on each type of medium. Fungal cultures need to be delivered to the lab promptly because one of the issues we run into with fungal cultures is that many of them do come from parts of the body that are normally colonized with many bacterial species. If there's a delay in delivering the specimen to the lab, if hours elapse, for example, these bacteria can actually overgrow any fungi that were present in the original specimen and inhibit the fungal growth. So we may miss a fungus on our plates if the specimen sat around too long before it was cultured, simply because the bacteria have outgrown the fungi. I will say that in terms of these skin scrapings, um, some doctors, unfortunately, don't do a real good job of getting them. I don't know if it's fear of hurting the patient or the way they were trained or whatnot. We do need more than just the epidermal layer, the dead skin cell layer, and sometimes they have a tendency to just kind of flake off some of the external layers, and that's not a good specimen. Dermatologists are notable for getting good skin scraping specimens. They go into the dermis, which means they're going to draw a little blood and get us a nice thick specimen that is much more likely for us to be able to recover a fungus from. Specimens arrive in the lab. Besides setting up culture media, we're going to do typically at least one or two direct exams. The particular direct exam that is done depends on the body site where the specimen came from. Also depends, of course, on the protocol that is present in the particular lab that you're working in. So one thing we can do is just a wet mount with some saline. And for some specimens, this will do it. We drop a saline on the slide, a little bit of the specimen. Try to break up the specimen as much as you're able to with a teasing needle. Put a cover slip on it and see if you can spot the various fungal elements. A way to kind of beef up a plain saline wet mount is to make a wet mount using something called lactofino cotton blue. This is a blue liquid, and when we suspend the tissue in lactofino cotton blue, what happens is If there are fungal elements present, they tend to take up the blue color more than just, the background blue shading and make things a little more visible, a little more distinct. So a little easier to find, number one, and a little easier to examine closely. When we get specimens that are consist of skin, hair, or nails, there's a lot of protein in that specimen. Talked about the skin scrapings in the previous slide. Sometimes the specimen will come down as several hairs that have been plucked from somewhere on the person's body. Sometimes it'll be a fingernail or a toenail that's been clipped off. So, in order to see any fungus that might be there, we want to get rid of that protein, that keratin primarily. And so a nice way to do that is to make a KOH prep, potassium hydroxide. So a little bit of the specimen on a slide, a drop of KOH. And the procedure differs a little bit depending on where you're at. Some procedures may say cover slip it and let it set for 10 minutes. Some procedures may have you cover slip and then warm it a little bit to kind of speed up that incubation time. The potassium hydroxide has the function of dissolving the keratin. And so we get rid of a lot of background material by doing this. We kind of free up any fungal elements that might be there and make them much more visible for us. But the key thing with the KOH is that waiting period. Sometimes we're in a hurry and we want to slap specimen on the slide, add a drop of KOH and go for it. That won't work because it does take a certain amount of time for the potassium hydroxide to dissolve the protein. Heating the slide a little bit speeds up the process, but there still has to be a waiting time. And kind of one of the drawbacks of having the specimen heated is we all kind of have a tendency to overheat and boil this thing because it'll boil very quickly. And then that kind of destroys the integrity of the specimen and you have to go back and start over. So sometimes it is best to just set it on the bench, set a timer for the prescribed period of time, and then go back and look at it. And lastly, sometimes we do a gram stain. Now, typically, if it's just a fungal culture, an examination ordered, we don't do a gram stain. Just because the KOH, the lactophenol cotton blue procedures are more sensitive and we're more likely to pick up a positive that way. But often, if the doc has collected a specimen, he's going to order a fungal culture and a bacterial culture because he doesn't know what's going to be the causative agent. And so with the bacterial culture order, we will have done routinely a gram stain of the specimen. So it is possible to pick up fungi via the gram stain method. We tend to call fungi as gram positive, and I just want to point out that's a little bit of a misnomer. If you're familiar with the gram stain procedure, the crystal violet adheres tightly to the thick peptidoglycan layer of the gram positives, and then it doesn't decolorize very well, so you end up with a blue color when you're all done. The reason I say technically fungi is not gram positive, there's no peptidoglycan in the cell walls, but the composition of the cell wall is such that it will retain the crystal violet, and typically they do end up the blue purple of a gram positive organism. I will say that yeast, for example, like many gram positive bacteria, as they get older, they tend to over decolorize a little more readily. Some of them, depending on the species, will decolorize more readily from the get-go. And when you get very experienced and you realize, wow, this yeast really over decolorized really easily, that gives me a hint that I want to see, if it's a particular yeast that I have in mind. A dead giveaway, whether you're looking at yeast or gram-positive cocci, is that yeast are always going to be bigger, at least a couple times bigger than a gram-positive coccus. If you're a microbiologist, you think about micrococci, which it's kind of weird. They're micrococci. These are gram-positive cocci, but they're actually big ones, typically on the gram stain. So I don't know where micrococcus came from in the name. But anyway, micrococci tend to be large, gram-positive cocci on a gram stain. Yeast are even bigger than those. You may see hyphae. Um, Sometimes they kind of mimic gram-positive bacilli to a certain extent, but again, with experience, you'll recognize that these strand-like structures are going to be wider than any gram-positive bacillus that you're accustomed to seeing. They don't always stain real reliably either, and they may have a much more irregular shape than a gram-positive bacillus would. Other stains that are routinely used, maybe a little less often than the previous ones, but other stains available for fungal examination: the acid fast stain, the India ink, and the calcifluor white. The acid fast stain, um, often associated with the mycobacterium bacteria. These are the ones that cause tuberculosis and some other illnesses. There's a fungus called nocardia. The genus is Nocardia. It is acid fast, just like those mycobacteria are, if you happen to be familiar with them. So, anyway, if the doc is suspecting nocardia, if we have reason to suspect nocardia based on, the specimen type, or let's say this organism has grown out on our plates, and we have reason to suspect nocardia, we may elect to do an acid fast stain to test for that property. India ink is something that you have to go back a number of years. Many of you will not have heard of nor seen India ink, but back in the days when people used ink pens, um, the ink that they dipped their pen into was commonly called India ink. And it's just a very, very dark black ink. And in the microlab, it's very useful if you suspect a yeast that is encapsulated, a yeast that has a capsule around it. And we'll come back in a later lecture and talk about a species or two of yeast that is encapsulated. And so the India ink procedure can be very helpful in determining whether a yeast has a capsule. And then lastly, you may have available to you something called calcifluor white. The most useful thing about calcifluor white is it's a fluorescent stain. And so if you have a fluorescent microscope, doing the fluorescent stain is a marvelous technique to have at your availability, because makes the staining procedure much more sensitive, much more easier to detect even small numbers of organisms that might be in a specimen. As with bacterial cultures, there's a number of types of media available, not quite as many for the fungi, but there are different ones that have various uses. Many fungal cultures will initially be plated on something called Sabarodes dextrose agar. The Sabarode part of the name comes from Dr. Sabarode, who developed this particular formulation years ago. And you can see it's quite obvious that the sugar source in this is dextrose. We often abbreviate Sabarodes with just SAB. So you may see a Sabarodes, with brain heart infusion. This is for enrichment to help grow some of the more fastidious fungi. And then sometimes we have SABs with brain heart infusion and blood added as well. Again, to add in the enrichment of the media. When we subculture fungi, in other words, we've got growth on our initial media, Some common ones that we use would be what we call just an Emmons sab. It just isn't quite as enriched, but once we've got this thing growing, it's just kind of a general purpose plate. It's kind of like a tryptic soy agar plate if you're doing bacterial cultures. It's kind of just a workhorse. Potato dextrose agar has some very specific applications. We'll run into those down the road. There are also several media that are available for fungi that besides having enrichment substances in them, they also have various antimicrobial agents. And these are specifically in there to inhibit the bacteria that might have been in the specimen, again, to allow the fungi to grow up. Sometimes they contain agents that are actually antifungal agents, but their purpose is to inhibit the rapid growing fungi. As we're going to learn, most of the rapid growers are not particularly pathogenic, and the pathogenic fungi are often much slower growing. So sometimes we actually have an antifungal element in a fungal media to cut down on the rapid growers to give the slow growers an opportunity to grow up for us.